Hello, my name is Sophia Greta, and my presentation is about the relationship between the microbiome, obesity, and also the brain. This presentation will focus on two key studies. We'll explore how gut bacteria interacts with the brain and how it can influence obesity. Obesity is a global health issue linked to conditions like cardiovascular disease, diabetes, and cancer, and it affects millions worldwide. So, why is this topic considered cutting edge? Researchers have now turned their attention to the brain gut microbiome, or BGM axis, to better understand obesity, which may give us a greater understanding of the condition of obesity. This emerging field shows how gut bacteria may influence brain activity and eating behavior, which can help us develop new insights about medical nutrition therapies. The first study is titled A Distinct Brain Gut Microbiome Exists for Females with Obesity and Food Addiction and was conducted by Dong et al. In 2020. The research question is, how does food addiction, or FA for short, correlate with the BGM axis in females with obesity? The variables the study measured was gut bacteria composition, levels of neuroprotective metabolites, and brain reward network connectivity. This study involved 105 female participants categorized by food addiction using the Yale Food Addiction Scale. As for the data collection, researchers collected stool samples for microbiome sequencing, analyzed metabolites, and used brain imaging to measure connectivity in reward regions. The results revealed significant differences in gut bacteria between food addiction and non-food addiction groups. Specifically, individuals with food addiction had higher levels of megamonas and lower levels of Akkermansia, Eubacterium, and bacteroides. This gut composition suggests that gut bacteria may influence overeating and therefore weight gain. This has implications for research about personalized nutrition recommendations targeting the gut. In addition, brain imaging showed stronger connectivity in reward regions like the putamen and the brainstem, driving overeating behaviors. This data suggests that disruptions in the BGM axis may underlie obesity-related eating patterns. This study had strengths, including its comprehensive integration of microbiome, metabolimics, and brain imaging, as well as its novel focus on studying food addiction, which is currently lacking research. Also, it provides sex-specific insights by studying only females. However, this small sample size lacks findings on men and limits generalizability. It also has a cross-sectional design which prevents causal conclusions. As well as in the analysis section, there was limited dietary control. Moving on to the second study, which is titled Obesity is Associated with a Distinct Brain-Gut Microbiome Signature that Connects Prevotella and Bacteroids to the Brain's Reward Center by Dong et al. In 2022. This is also the same researcher who conducted the first study we reviewed. The research question is, what are the differences in brain-gut microbiome, or BGM, characteristics between obese and non-obese individuals when controlling for diet, race, and sex? The variables that were measured in this study was that Prevotella and bacteroids ratio, nucleus accumbens, or reward center activity, and fecal tryptophan levels. For reference, Prevotella has been implicated in certain inflammatory conditions when present in excess, whereas bacteroids is generally seen as more beneficial in maintaining gut homeostasis. This study included 287 adults, 99 males, and 198 females that were either classified as obese or non-obese. Researchers used fecal samples for microbiome profiling, dietary questionnaires, and resting state functional MRI to measure brain connectivity. Now, let's look at the findings of this study. Obese participants had a significantly higher Prevotella/bacteroids ratio, or PB ratio, compared to non-obese individuals. If you look to the right, we can see that in the non-obese individuals, they have higher amounts of bacteroids than Prevotella compared to the obese groups. An elevated PB ratio may point to an inflammatory microbiota profile, which could influence metabolic diseases like obesity. People with obesity were also found to have lower fecal tryptophan levels, which reduces serotonin production. This is important because serotonin regulates appetite by promoting satiety and suppressing overeating. Brain imaging revealed that higher PB ratios correlated with increased activity in the nucleus accumbens, a reward region linked to overeating behaviors. These findings suggest that PB ratio and nucleus accumbens activity could serve as biomarkers for obesity. This study's strengths include a large, diverse sample size, detailed microbiome analysis, and the use of brain imaging linking gut bacteria to reward activity. Its limitations include that it's a cross-sectional design, which limits causal conclusions. It also relies on self-reported dietary data and lacks long-term analysis. In conclusion, the key findings highlight the role of BGM axis in obesity. The first study shows that food addiction is linked to gut microbes and brain connectivity, while the second identifies the PB ratio and brain activity as potential obesity biomarkers. In addition, these findings have practical implications for dietetics. Microbiome-targeted interventions, such as recommending a diet higher in probiotic foods, could help manage obesity. Also, biomarkers such as PB ratio might guide personalized nutrition strategies. Future research should focus on the long-term effects of microbiome modulation and the diet on obesity outcomes. Expanding these findings can enhance medical nutrition therapy. In future clinical practice, dietitians could use microbiome-related biomarkers as tools to optimize medical nutrition therapy for obesity management. In addition, we may be able to one day make more accurate nutrition recommendations for specific foods based on research about specific microbes. And here are my references, and thank you so much for listening.